So we are switching gear a bit today, and today we will still talk about consciousness, but this time we are talking a lot more about speech and language, which is the main topic of day 2. And we have a number of fantastic speakers. So our first speaker today will be Professor Ying Mao from Fudan University. And I just noticed you are a neurosurgeon by training.
Yes.
As you may know, when I studied medicine, I was toying for a while with the idea of becoming a neurosurgeon myself. And before I switched gear and went into neurophysiology, I did even time of training. I went to Harvard University and did some neurosurgery. And when I see what you've been doing in the meantime, I say there's quite a bit of overlap between the hardcore basic sciences and the neurosurgery and the application to this. So I'm really looking forward to Dr. Mao's talk today. And the title of his talk is Machine Learning in Clinical Neurosurgery and Applied Neuroscience. Dr. Mao, the floor is yours.
Okay, thank you. I just share my screen. It's okay?
Looks good.
It's good, okay. So good evening, ladies and gentlemen. I'm Ying Mao, a neurosurgeon from Huashan Hospital affiliated to Fudan University. First, I would like to thank Science and the TCCI for co-organizing such cutting-edge research in cognitive science. We had very fascinating talk on day 1, and I'm grateful for the opportunity to present some of our team's work on the application of the artificial intelligence algorithm in the diagnosis of neurosurgery and neuroscience research. The subject of my presentation is machine learning in clinical neurosurgery and applied neuroscience. The essence of the machine learning is consistent with the learning model of human brain, depending on the accumulation, application, and modification of the experience. As we all know, the doctor was quite familiar with this procedure. The training and groups of doctors requires the study of a large number of the cases and absorption of the new information so that they could make decisions more accurately. Otherwise, as we said, garbage in and garbage out. Machine learning is not equal to artificial intelligence. In fact, machine learning is only part of the artificial intelligence, but it's indeed the most important part. Deep learning and reinforcement learning, which are currently very popular nowadays, belong to the category of the machine learning. The application of the machine learning in the medical field had a long history. The birth of the first computer-aided diagnosis system in 1966 was the prototype of the application of the machine learning in medical diagnosis. By 1976, the expert database and the IBM Dr. Huashan robot was all models of the application of machine learning in medical diagnosis. The application of a machine learning has developed relatively late in the field of the neurosurgery and the neuroscience. Since machine learning simulates the working mode of the brain, these two fields was precisely need the help of the machine learning. Meanwhile, the machine learning provided an accurate method for analyzing the massive information of the human central nervous system. The accuracy and the stability of our algorithm provide the possibility for subsequent use in brain surgery and brain research. Therefore, the application prospect of the machine learning in neurosurgery and in neuroscience are very broad. With the development and the evolution of the machine learning algorithm, neurosurgery has been developed from the earliest general neurosurgery to digital neurosurgery, and finally, intelligent neurosurgery, which we called a new generation of the neurosurgery. And machine learning technology has also been widely used in medical education, disease diagnosis, preoperative planning, intraoperative navigation, and postoperative management. For example, the traditional craniotomy is time-consuming and risky. In the future, neurosurgery equipped with the next generation of neurosurgical intelligent robots will be very efficient and easy in surgery. Although machine learning is currently used in many industries and has achieved a certain commercial success, we must realize that the machine learning we use today is still so-called weak artificial intelligence, rely on a large amount of high-quality data for training and validation. Based on the massive medical data of the Department of Neurosurgery of Huashan Hospital, the content of my speech today will share some of our team's work on machine learning application in neurosurgery, diagnosis, and treatment. I will introduce the machine learning in three aspects: the medical imaging processing, EEG analysis, and the surgery assistant. The first part is about the medical imaging processing, in other words, radiomic study. Radiomic study is now very extensive. Utilizing imaging information can assist the doctor in diagnosing disease, predicting prognosis, and also formulating treatment plans. The imaging segmentation and feature extraction and classification prediction are the basic analysis method of the radiomic study, which are also classical application of the machine learning in the medical field. Pathological images are the most commonly used medical imaging and directly affecting the diagnosis of disease. Therefore, we first use the deep learning CNN network to identify pathological images and develop the automated pathological diagnosis system for glioma. This is the flow chart for this automatic diagnosis system, which we called AR neuropathologist. By scanning the pathological HE staining section into the computer, and this system can provide histological and molecular subtyping diagnosis of this section based on the previously established deep machine learning model. This video shows how
Pathology is
AI
the golden
neuropathology
standard for
works.
cancer diagnosis. So, we have developed an AI pathological diagnosis system for brain tumor. Those are the slides for pathologists to diagnose. We placed the slide on the microscope and used the highest magnification to align the bottom left of the slide. The view under the lens is transferred to screen by the camera on top of the microscope. Then we click on Start Scan. The micro-positioning platform starts to move. This movement is almost undetectable. The platform moves in an S shape, driven by the control box alongside. 300 local images of the slide are taken and saved. Then we open the image assembling software and select all the images. All the images have a 30% overlap with its surrounding ones, which helps with the assembling process. Now we shall have a completion of the slide. Next, open the diagnostic software we developed with a GPU server and run the program. A histopathological diagnostic report is displayed on the screen within a minute. The model is built up by more than 90,000 images of 300 confirmed cases using deep learning. with an accuracy over 85% and will be improved by the increasing number of cases. Finally, enter molecular information. According to World Health Organization CNS Tumor Guide, the final diagnosis will be given through logical derivation. Our system would assist pathologists with better diagnosis.
So after completing the diagnosis of pathological images, we then use the machine learning to analyze the magnetic imaging, MR images. MR imaging is the most important basis for the diagnosis of brain disease, which is also non-invasive. So the quality of the MR images obtained with the different instruments and the imaging parameters varies greatly. Such imaging quality difference will have the great impact on the radiomics analysis. Therefore, it is necessary to resolve the consistency problem of the MR images. The main difference in MR images, including the modality mismatch, intensity distribution variance, and the layer spacing difference. So for intensity distribution standardization, we just established the cycle gain model. The cycle means there is a generator of the target imaging to the reference image, another generator of the reconstruction imaging to the target image. The purpose is to make sure that the target imaging passes through the two generators and was still consistent with the original target imaging. By using this model, we just can adjust the MR images from different hospitals and different scanner to the same intensity. So this is just the examples. The step 2 is that we just designed the two GAN network for the layer spacing normalization for infant and adults, respectively, which means no matter which layer space did you use, we can normalize them into the certain layer spacing. Usually we turn imaging with thick layer spacing into ones with the thin layer spacing. Here is the example. We just transfer the layer imaging to thick layer images. So certainly, the MR have hundreds of modality. If the modality of the enrolled case is inconsistent with other cases, or if the certain modality is missing, this case becomes useless in radiomics analysis. We solve the problem by missing modality reconstruction. Here is the examples. We use the T1 and plus T2 modalities to reconstruct the T2 flare modality. After the imaging standardization, we just established a 3D unit segmentation model for lesion segmentation. The performance in segmentation is good. The dice number is 0.8. And this segmentation of one modality MR can be completed within a couple of seconds. And this is just a multimodal segmentation can be also achieved by using this 3D unit model. When we finish the first and the second step, we can use MR radiomics data to realize the non-invasive glioma molecular diagnosis. The accuracy is almost the same as the real molecular testing. Meanwhile, we can find the intra-tumoral heterogeneity inside of the tumor and do the precise risk stratification with the same glioma subtyping. Recently, we just constructed a new research on radiomics. We combined the HE and MR data to establish we called micro integrated analysis model. By using this model, the accuracy to predict temozolomide sensitivity can reach 90%, higher than the single modality radiomics method. And this is our AR glioma precise diagnosis and prediction system. We are carrying out multi-center clinical trial to validate the efficiency and the accuracy of this AR system. Moreover, the machine learning can also be applied to the diagnosis of the cerebrovascular disease. We have established a set of the 3D CNN algorithm to diagnose Moyamoya disease and predict the risk, the breeding risk of the Moyamoya disease. A total of 612 cases was included in this retrospective study. Our model has an accuracy rate of 0.9 for predicting the outcome event of breedings of the Moyamoya disease. Based on this algorithm, we just developed an APP. By importing the DSA imaging data, this APP can determine whether the patient had Moyamoya disease and predicting possibility of the breeding risk related to the Moyamoya disease. So this is just the a show about of the whole procedures. In the second part, I will introduce the application of the machine learning algorithm in EEG analysis, especially the role of mapping brain functional area and decoding the neurocircuit. In addition to EEG, there are many methods for the brain function research, including the functional MR, PET, combining the spatial and the temporal resolution. ECOG and the SEEG are the best parameters reflecting the brain function. And direct electrocortical stimulation based on brain mapping techniques is the gold standard for the intraoperatively delineating eloquent area. But direct electrocortical stimulation meets its burden because of the time-consuming and high risk of inducing the seizures during the awake surgery. Thus, we discovered an automated procedure that complements routine intraoperative medial nerve stimulation to rapidly localize the central circles and to create the functional somatotopic map, which while the subject is out of risk. This is what we call the passive brain mapping. We first developed the algorithm that automatically localized the central circles based on the phase reverse technique by analyzing the medial nerve stimulation evoked somatosensory evoked potential. We call it the SSEP. And the sensitivity and accuracy is correct with the conventional electrophysiological method. Next, we just started to use the MNS-induced response to create a functional map of sensory motor cortex. Not only use the medial nerve stimulation, we also use the vibrotactile stimulation to constitute a subset of the somatotopic map generated by our functional mapping techniques. Interestingly, our results find out that the broadband gamma response to MNS and vibrotactile stimulation while subject was awake, and broadband gamma response to medial nerve stimulation while subject was awake or under deep sedation. This is just one case of recent awake surgery. After 2 minutes of the medial nerve stimulation, we can immediately get the result or predict the motor and the sensory cortex. The yellow line indicates the central circles, and the yellow and green dots represent the motor and the sensory cortex. Then we performed the traditional way of mapping. H means hand motor, S is for shoulder. The result was practically identical, and the new method can show the sensory area while the direct cortex stimulation cannot. Furthermore, performing the mapping when the patient was awake is still not ideal. We tried mapping during the deep sedation state. In this case, the result was the same. The high sensitivity of 87% and the specificity of 86% of attenuation during the deep sedation as the index for the functional related area. Localizing the motor cortex is the first step. we need to know the motor-related cortex activity sequence. This will help us precisely protect the whole functional pathway. We made the investigation on SEG patient. This study was approved by the ethical committee of Huashan Hospital, and all the subject participants a visual cued finger and arm movement task. We first analyzed which area is responsible for the visual cue task. We found multiple regions was activated during the task, and the motor cortex, occipital cortex, and the parietal cortex are among the most significant ones. Meanwhile, we developed a set of the algorithm to identify the time order of different brain regions during the motor task. We applied the task starting time and the EMG activation time as the reference to normalize brain activation time. In our result, we can see the activation sequence of the different region. Each brain region is color-coded, and the number presented the order of the brain activation, and occipital lobe come first, and central lobe the last. This video just shows the neural activation sequence on the large scale during the visual cued hand motor task. The colored brain area indicates the neural activation in this area. At the final stage of the motor execution, the precenter area produced a motor signal from already highly processed sensory input and the other internal signal. The postcentral cortex produced the somatosensory feedback after the motor execution. From this result, we can see fine motion decoding and high quality signal processing was very important support for the brain computer interface, which means that the BCR. Well, we just developed a machine learning algorithm in the basic neuroscience research. In this zebrafish and the paramecia study, we used the DNA to achieve the automated tracking. The paramecia and the eye tracking result was showed for the 4 different haunting sequence corresponding to success, abort, miss, and reject. And the yellow circle denoted the position of each DNA tracking paramecium. The blue and the red dots show the 4 DNA tracking eye position. The description on the top right corners, the eye convergence, which in the red, and eye unconvergence, which in the cyan, indicated the eye convergence annotation determined by the eye tracking result. You can see that the different tasks can have the different color. And simultaneously, we also recording the whole brain neural activity to decode the zebrafish specific behaviors, which is a classic method for the brain science research work. So in the third part, I would like to discuss the applications of the machine learning in neurosurgery. For neurosurgeons, neuronavigation is the GPS of the surgery, and also it's the promise for the success of the many neurosurgical operations. Therefore, our team has been committed to researching how to use the machine learning method to achieve the automated surgical navigation. Intraoperative MR is the gold standard for the neural navigation accuracy. However, the intraoperative MR is expensive and time-consuming. Considering the advantages of the easy operation and the low price, ultrasound will be the ideal alternative tools. However, since many doctors was not trained to interpret ultrasound imaging, it is difficult for them to understand the real-time ultrasound imaging professionally. So in order to solve the problem, we recently launched a study on intraoperative ultrasound navigation. We previously collecting the ultrasound imaging of the tumor during the surgery, let the sonographer make real-time diagnosis. Though through the establishment of the deep learning model, the result was fed back to intraoperative navigation. And this model can directly mark the tumor with the high brightness, therefore helping neurosurgeons to carry out the real-time intraoperative navigation. So this technical method is also expected to solve the problem of intraoperative brain shift and saved a lot of operation time for the neurosurgeons. Similarly, the machine learning can also be applied to the simulation and the formulation of the interventional surgery plans for the aneurysm. After completing the DSA, the model can analyze the aneurysm and also the aneurysm-bearing blood vessels and simulating the microcatheter treatment plan. We use the 3D printing to shape the microcatheter and optimize the efficiency of the embolization. This is just the case for the carotid ophthalmic aneurysm, and we are going to perform the interventional embolization for the patient. Use the machine learning and the 3D shaping method, we can formulate the most optimized microcatheter treatment plan for the interventional embolization, and the effect of the patient treatment is satisfactory. It's similar to the previous report method in zebrafish and paramecial visual tracking. We just apply the improved cell-band deep learning network to continuously track the intraoperative target field of the view. Combining with the self-developed voice-controlled exoscope, we visualize the rear vision navigation during the operation. And this is just an example. No matter how the lens moves, or there is a bleeding around or obscured by the surgical instruments, this method can ensure the correct tracking of the surgical field of the view, which is a great help for the neurosurgery. So in the end, I'm going to summarize the role of the potential problems of machine learning in neurosurgical applications. The machine learning method provides neurosurgical friendly medical tools in the potential clinical practice, for example, the diagnosis and treatment, and also prognosis planning. Having things simple and let go faster and making decisions right are the core of the machine learning in neurosurgery and in neuroscience. And also the multi-center large sampling prospective clinical trial was essential for the machine learning based method to be used in the future clinical neurosurgery. And also the black box should be unveiled to prove the authenticity of the machine learning based medical tools. And also the more intelligent machine learning based method need to be developed, and also be friendly with the machine learning and trust yourself. So just like I mentioned previously, right now, the machine learning technology is just a weak to medium artificial intelligence. There's a long way to go to fulfill the goal of the intelligence neurosurgery. Like mountain climbing, we can only see the short distance ahead, but we can see plenty there that needs to be done. Thanks to my teams and collaborators, our smart neurosurgical research have got support from a lot of foundations, including TCCI. And this is my whole lectures, and thank you for your listening. Hope you enjoyed it. Thank you.
So, thank you very much. A brilliant overview. And you say that we have quite a long way to go, but from what you presented is we have already gone quite a bit. So there has already been quite a lot of advance over the last years. So I'm seeing that there is a lot of questions coming in. So let's start with some of them. The first question is pretty straightforward. Does machine learning perform better than clinicians when it comes to diagnosis?
This is a really good question. That right now the deep learning or the AR diagnosis is still based on the clinician's experience. So right now, so we try to let we just have established a very big imaging data bank and try to teach the machine to learn more and more the actual clinical cases. But I think that right now, so we still depend on the feelings of the clinicians. So I think that it's maybe still have a long way to go to have the real AR diagnosis system to replace, or actually it is not possible to replace the clinician's experience.
Okay. Here's a question, this is more general. The signatures derived from the image, how can they be connected to the gene mutation status? Could these features be readable for humans?
Yes, this is also a really rare question. The purpose of the neurosurgery is that we really want to understand or to realize the molecular diagnosis of the patient before the surgery. So that's why we just connect we just have more than 1,000 patients, and we just have the MR data to be studied by the machine, and we just have the molecular testing of the all cases and try to feed the status data to the machine and try to let them understand some of the parameters for the submolecular for the molecular diagnosis. And then finally they just have the radiomic diagnosis. So I think that in the future, so we can find some parameters on the MR scan and to understand that this is just some presentation, there's some ideas about the molecular informations. So I think that right now so we can have the accuracy of 90%. So this is it's a really high of accuracy for such kind of study.
Here's a question that I may not fully understand. Under what data set assumptions that GANs is applicable, and under which circumstances can it be not used? For example, different distributions, out of distributions, and how to deal with the missing modality in real world application.
Yeah, as we know, the MR scan, the information what we got from the MR scan is totally different from the different machine and the different sequence. So that's why we just try to normalize all the images. This is a basic for the machine learning. So that's why we try to suppose that in the different slides, the consistency of the tumor is still there. So that's why we can use the reconstruction techniques and try to reconstruct the images which is suitable for the machine learning. But actually, this is artificial imaging. This is not the real imaging from the true patient. So this is maybe it takes a time to try to reconstruct the images and try to normalize it. So I think that it's not actually it's not very accurate right now, but we still think that this is the target and the future. We can use such kind of technology to predict to subtraction the lesions from the imaging.
So here's a question. What is the effect of 3D spatial temporal labeling for the classification accuracy?
Actually, we in the neurosurgeon springs, we may have a 3D structures of the brain and also have the 3D structure of the tumor. But actually, what we got from the MR scan is just the 2D images. So we try to use the this technology and try to reconstruct the 3D structures of the tumor, so that we can accurately analyze and predict the growth factors, kind of the tumors.
What are the evaluation criteria used in the multiple target tracking? And how far is this from practical application?
The multiple, the method, it's, I think that this question is really hard to understand, to answer, because I'm a neurosurgeon, and actually this is done by the scientists from the computer science.
Can you give some common problems currently at AI? Are there any specific problems for the application of AI in neurosurgery?
I think that this is a lot of advantage for the AR used in the neurosurgery, because the brain is fixed, and also the structures of the brain is also fixed. So that's why we can use such kind of advantages, and we can have the AR technology to understand the structures of the different robe. So I think that the AR can be used in the neurosurgery clinical work in the very near future. And actually, right now we use a lot of technologies right now in the neurosurgery. And I think that in the next speakers, the Professor Chang will just talk about more about the technology used for the speech technology. Yeah.
How do you ensure the external validity of machine learning research?
Okay, so this is also a very good question that right now we just have a lot of we're just collecting a lot of patient data, and also we just used such kind of technology and try to validate in the real surgery. This is something like the prospective study, and we use the neural navigation system and try to use the biopsy to underlie to confirm the border of the tumor, and also sometimes we just find some significant changes on the imaging and try to use the biopsy to confirm that in the real surgery.
Now we are going further into surgery, and someone has a problem with or a concern about the safety of assisted surgery equipment. What would we do if the machine learning assistant scope consistently provides us with an inappropriate surgical view?
Pardon? Excuse me?
Yeah. I'm repeating the question.
Yeah.
This is a question about the safety of the surgery equipment. What could we do if the machine learning assisted scope consistently provides us with an inappropriate surgical view?
Mm-hmm. I think that actually right now the neurosurgery right now has become mini invasive. So sometimes we just use the biopsy, just burn a hole on the bone and try to have the biopsy. So that's why we really need the AR try to outline the whole structures and whole pictures of the brain so that we can understand the pathway for the neurosurgical, for the neurosurgeons to do the surgery. So I think that it's very important for the neurosurgery because we could not open the whole brain. So we just use the burr and try to understand and to realize the whole brain structure. So the AI can make the surgery much more safer than before.
So now we have a question. This is more a general question about the present state of machine learning. As you mentioned, at the moment we are at a stage of weak AI. And this means we need to input a lot of high quality data to let the machines learn. And then a mode can be set for further utility. So the question is, how can we achieve good machine learning results?
So I think that the high quality data, especially from the MR, is very important. So we have to feed more and more the imaging data and also some experience from the neurosurgeons. So I think that with the development of the AI technology and also the accumulated more and more clinical data and also the image data, I think that the computer become smarter and smarter. So I think that I just have a confidence that in the future, in the near future, we can use such kind of the AI to help the neurosurgeon. But honestly, it is not possible that we use the machine, the robot to replace the neurosurgeon. So we still can control the surgical procedure.
Along the same line, the question here, how can we overcome the limitation? Most of the time we have a limited data set. So sometimes we may just have a data set obtained from a single medical center or a single ultrasound device. So you must have been thinking about this.
Yes, so I think that this is just the advantages in our department because we have more than 700 beds for the neurosurgery. And also we have more than 200 neurosurgeons worked in the same clinical centers. So we have more than 20,000 craniotomy every year. So I think that still we need a multi-center clinical trial to have more and more clinical data to support the findings from our department. And I think we are just launched such kind of the multi-center clinical trial with the support from the TCCI. So I think in the future we can have a very accurate data.
Yeah. Along the same line, here's a question about acceptable sensitivity and specificity in different clinical applications. And this is a problem that's probably not only for neurosurgery, but I think this is very widely used. How can we avoid potential risks caused by false positives and false negatives?
Yes. So I think that it's just aided by the real clinical surgery. And we right now, so we just use this such kind of the AR aided system, but we still just keep in mind that the neurosurgeon should control the surgery and try to use all kinds of the chance to confirm and validate the result from the machine learning system. So I think that still I think that high quality data accumulated is very important for the machine learning development.
So someone is focusing on the ultrasound imaging. Yeah. What kind of images should we use for making predictions? 2D cross-section images, 3D ultrasound, or perhaps even 4D spatial temporal data?
This is I think that we are just in the very early stage for application of the ultrasound in the surgery, and also it's really early because right now we use the 2D imaging getting from the preoperative imaging just after the craniotomy. So because we can avoid the inference of the bone structures. So the images from the brain, the real brain, after the craniotomy is very clear, but it still have some distortions. So that's why we just let the sonographers to interpret these structures. So it's really hard for the neurosurgeon to understand the anatomical structures got from the ultrasound.
So still along similar lines, people seem to be worried quite a bit about accuracy. How high has the accuracy to be? How high has the accuracy to be considered clinically useful? And in particular, in terms of neurosurgical practice, because this is such a highly skilled feature where you're really dealing with minute changes.
Okay, this is a really good question. Actually, we don't know what the actual number for the accuracy is can pass the permission for the clinical application. But I think that 95% of the accuracy, sensitivity, and specificity is maybe it's the final stage. But actually, I think that the brain surgery is totally different from the other surgery because the old structures in the brain is fixed. So that's why they offer us the good chance to use such kind of technology, and we can easily to got the very high sensitivity and the specificity.
There's a question on brain-computer interfaces. Could they get higher quality processing to help the diagnosis? And do you, in your hospital, have made specific progress?
Actually, right now, the problem for the BCR is that we try to collecting the very clear signals from the brain. This is the first stage. The second one is try to analyze, or we call this decoding, the signals got from the electrodes. And also we try to modulate the brain. So I think that right now we are just at a very early stage. I think that right now we are very happy that the Edward Chang just have a very good result get for the BCR. I think that we really have a very detailed study about the functional areas in the brain. So right now in China, we try to use the similar technology, and we use the similar the electrodes, for example, the Utah electrodes, to collecting the signal and try to analyze it. So I think that we try to learn from the Edward Chang and try to learn how to analyze the neural circuit in detail.
So now we are coming towards the end, and there are a few more general questions. You mentioned more than once the black box. So how important do you see the role of explainability of the AI models in your work? And are there pathways to improve explainability?
Yes, this is a really good question. For the doctors, we really want to know the mechanisms of the artificial intelligence. And actually, right now they have different models, and we have no idea about such kind of the models. So what we can do is try to validate the result from the different kind of the model got from the computer scientist. So I think that this is called the black box. And I think that this is still it's not we call it it's artificial intelligence, but actually it's not the real intelligence from the human brain. So I think that we have to validate that in the future.
Yeah, talking about the future, there's someone with a very broad question. What is your vision of the future of neurosurgery in the context of healthcare systems in 20 years from now?
I think that the neurosurgeons are very smart guys, and they try to study from the different department, for example, from the cardiologist and from the nephrologist. But actually, they are trying to have the minimally invasive surgery, because right now we call it a general neurosurgery. We have to open the brain, have the craniotomy. But I think that in the futures, we can just burn a small hole, and even sometimes we can use a very tiny pathway and try to put the catheter, put the electrode inside of the brain and try to modulate it. I think that in the future, the surgery become less invasive or maybe non-invasive. That's the future.
And along the same lines, we're still talking about the future. Would AI-assisted neurosurgery help reduce the inequality of medical resources? The questioner asks, for example, help for rural areas, help for developing countries?
Yeah, this is a very good question, because right now we try to use such kind of technology to control the robot in the rural area by the 5G technology. And the doctors can control the robot in the rural areas and try to do some surgery there, to try to educate and try to instruct the neurosurgeon there to do the surgery. And sometimes we can instruct the robot to do the surgery. But right now, so what we can do is that we try to use such kind of technology to implant the electrode in the rural area. So I think that in the future, so we can control the robot to and have the wonderful surgery in the developed country, also in the developing country.
Oh, thank you very much. I think on this optimistic note, let's come to a conclusion. And I'm clapping now, but I'm doing this for everyone who has attended the lecture. So you have really opened today's session with a brilliant overview.
Thank you.
Fantastic, yeah, thank you.